So as you just heard, my name is Max Spies. I'm a postdoc just up the road in archaeology under Prof. Seeley. And yeah, I'll be talking a little bit about our progress in developing a Strontium isoscape for the Western Cape and how we can use it to do a better job of reconstructing land use patterns in the past, in the Holocene specifically. So let me gauge how many people are familiar with Strontium. Like there's some familiar faces, but I don't know everyone here. So I wanted to see how much people know about this topic. But either way, I'll give a kind of broad overview of. Oh, thank you, great. I'll give a broad overview of what isotopes actually are, if you're unfamiliar, just in case you're unfamiliar. How we measure them, how that works, before giving some more detailed information specifically about strontium isotopes. I'll then go into how they can be used for provenance and estimating origin of archaeological samples, and as well as I'll touch on some other examples. Then I will chat about the current project, some archaeological context of the project, before going into how I collect and prepare samples and use them to develop an isoscape, which I'll explain a bit later. If you're unfamiliar with the term, an isoscape is a spatial predictive model of strontium variation, unsampled locations. But I'll get there. Yeah, then I'll share my results and discuss them a little bit and share some conclusions. So, what are stable isotopes? Basically, isotopes are atomic elements that have equal number of protons, but different numbers of neutrons. And that means they have the same atomic mass, but different atomic mass, but same chemical and physical properties. So the classic example is carbon, and at the top there, not really working very well. Anyway, at the top you can see carbon-12, which is made up of 6 protons and 6 neutrons. In the middle is carbon-13, which is 6 and 7. And then we have carbon-14, which is 6 and 8. And this is a kind of classic example, but isotopes exist in basically everything, all elements pretty much. So everything we interact with can have an isotopic various forms, including in our bodies. So there are also loads of different applications for isotopic work. Obviously, I'll be focusing on the archaeology tonight, but there are various other fields that use this kind of technology. How do we measure isotopes? Basically, it's pretty much impossible to weigh an atom, but using the fact that some of these isotopes are heavier and others are lighter, we can measure the ratio between the heavy and light versions. And we do this using mass spectrometry. So this is a little diagram. Of a mass spectrometer with an ion source at the bottom. It can't show up on the bright screen. Can you see my mouse? No, I can't see my mouse. Oh no. Okay, I'll just point. Ion source at the bottom, which is where you'd load in your sample. And that sample can be a gas or a solid or a liquid, depending on your machine. And it's then ionized by an ion, or first it's either combusted or vaporized, turned into a gas, basically, if it's not really a gas. Ionized by the ion source, which means it's given an electrical charge. And then these little charged particles are thrust down a vacuum tube into a giant magnet, which then bends the charged particles before they snap into a detector. And because they're somewhat heavier and the others are lighter, you can see that starts out as one kind of ion beam, And depending on the mass, because the magnetic field is the same and the charge is the same, the degree to which these ions bend depends on on their mass. So the heavier version will bend less because it's heavier. Because of inertia, it's going to bend less, and the lighter version will bend more. And depending on where they hit that detector, you basically get a count for how many heavy isotopes or versions of that element are in your sample versus how many of the lighter version. And then your computer will calculate a ratio of your heavy to light isotopes in your given sample. That's basically a broad overview of how it all works. And like I mentioned, most of the elements on this periodic table, if it comes back, there we go. Most of the elements have various isotopic versions. I am mostly interested in the element strontium, which is blue. And the reason for that is because it behaves quite similarly to calcium, which is its neighbor just above there. And that means it can replace calcium in biological systems. So that's one of the reasons why it's kind of interesting. So we find strontium in small amounts in plants and animals, and like in our hair and nails and bones and teeth and various other tissues. I don't know what's happening here.
No idea what's happening.
Coming back. Yeah. I don't think so. There's lights on the back there, so. No, don't do that. It's working. It's fine. It'll be fine. I think rather leave it. Patricia can't sit there the whole time. Go. It'll be fine. It'll be fine. Honestly. It'll settle. Okay. Sorry, everyone. Yeah. Okay, so as I was saying, pretty much all the elements on the periodic table here have isotopic versions. Like I said, heavier and lighter versions. They behave the same way, but they have different masses. I'm mostly interested in strontium, and like I said, behave similarly to calcium. And so we find it in human tissues and animal tissues and plants and things like that. Wherever calcium would naturally occur in a biological system, strontium can sneak in. That's one of the reasons why it's interesting. The other reason why it's interesting, I'm going to get to in the next couple of slides, but it's basically because it can be used as a provenance tool or a tool to estimate origin in any given sample, basically. So there are 4 naturally occurring isotopes of strontium. And we have strontium 84 at the top, 86, 87, and 88. And you can see that they have various abundance levels. Strontium 84 is relatively rare, and only about 0.5% of all strontium is strontium 84. 86 is close to 10%, 87 is around 7%, and 88 is the most abundant at about 82%. Specifically, though, we're mostly interested in the ratio of strontium 87 to 86. Those are the more interesting isotopes in the strontium world. And the reason for that is because strontium 87 is radiogenic. And that word basically means that strontium 87 is derived from the slow radioactive decay of rubidium 87. You can see rubidium 87 had a half-life of 49 million years, so it takes a very, very long time for rubidium to decay, but it does decay. And Rubidium and strontium are elements that are, like I mentioned, found in nature, and in particular found in bedrock in varying amounts depending on the rock. And so the presence of strontium 87 is dependent on the age of the rock, when in geological time that rock was formed, as well as the initial abundance of rubidium. I hope that makes sense. There's this kind of broad context that you kind of need to know in case you don't. So the important thing is that the ratio of 87 to 86 persists in the rock and in the soils that derive from those rocks, all the way up to plants and animals plants that grow in the soil, animals that eat those plants, humans that eat the plants and animals in that region. So your strontium 87 ratio, 87 to 86 ratio is kind of it represents a location. And that's how it can be used for estimating origin and estimating provenance. But bedrock is not the only source of strontium in the environment. And although it does have a dominant role, so most of the strontium in the soil comes from the rock beneath, there are other sources. And the rock itself, like different rocks, can age at different rates and contribute different amounts of strontium. So carbonate rocks versus silicate rocks, there's quite a big difference there. But they weather at different rates and contribute differently to the soil. Different plants will then take up that strontium and at a differential rate depending on their rooting depth. There are additional sources like sea salt and dust that's blown across a desert onto a different place. Volcanic ash is a big one. Now, various sources. Rivers can bring strontium from upriver down to a low elevation, so a different source down to a floodplain, for example. And then humans and animals will incorporate that strontium via their diet, but it depends on their feeding range. It depends on their dietary preferences. It depends on what's available for them to eat. So it's quite a complex system. It's not as simple as just understanding the geology. But basically, fundamentally, the ratio of 87 to 86 is characteristic of a location, like I mentioned. And this makes it a useful measure of provenance or origin. And in archaeological contexts, one can identify local versus non-local people or animals, maybe, for example. And you can do that by comparing a tissue from one individual to their environment, for example, or to different people within a larger group. Or one can also compare different tissues within one person and get an understanding of whether they moved around across the landscape, because different tissues grow at different rates, and they will incorporate strontium from the different areas where they moved, if they lived there long enough and ate food from that location. So there's lots one can do in an archaeological context. I also briefly wanted to touch on some of the non-archaeological use cases. So we'll have a quick sip of water. But in non-archaeological context, a cool example that I like is wildlife forensics. And there's work being done by SANBI, the South African National Biodiversity Institute, who are using strontium ratios to trace stolen cycads and succulents. So they're endangered plants that have been nicked from nature reserves, planted in private gardens. People claim to have had these plants forever. And you can test the root of that plant versus a new leaf of that plant, old growth, and get a picture of where that plant previously was grown, because it's not, it doesn't represent the soil that it's currently in. So that's a cool example. You can also trace poached animals like rhino horn or basically any wild animal. And there are some uses for modern human forensic tracing as well. I'm not going to go into that too much because there are some limitations, but we can chat about that later if people have questions. But some alternative non-archaeological examples, just thought it might be interesting. Let's move on to my current project and provide you with some archaeological context of what I've been doing. So over the last few years, around 50 human skeletons have been found along the coast, along the west coast. And these are the ones that have been dated using carbon dating. And they've been dated to between around 6,000 BP to 250 BP. It's quite a broad range. Obviously, this is Holocene stuff. And alongside the radiocarbon date, we have dental enamel strontium ratios for each of these individuals. And the important thing to remember about this is that dentition, your teeth, begin to develop in utero. So the strontium ratios of the individuals I'm going to be showing you in a couple of slides represent a strontium ratio that comes from the mother's internal strontium pool during gestation, as well as early childhood diet. So it's a mix of sources, but just keep that in mind as we go along. So during this period in the Holocene, South Africa was home to several hunter-gatherer communities with well-documented extensive use of marine resources. And this is a painting of such a community done by Robert Jacob Gordon in 1779, who met this group of people living on the beach. And in the foreground, actually, it's a fantastic painting. In the foreground, you can see lots of shells and whale bones, and there are various other things. That cloak is made from skins of penguins, I believe. Anyway, the point is good documentation of marine resource use. Alongside that, we have several large megamiddens, shell middens, that have been excavated along the coast, along the west coast. Like this one is from Paternoster. And you can see these are just layers and layers of shells, right? So this also represents long-term marine resource use, possibly multi-generational habitation of the same location, or at the very least, frequent reoccupation. So if people left, they came back because there's just lots and lots and lots and lots of shells. And then these top layers are dated to around 2000 BP, just for context. Later, though, also around this time, 2000 BP, with the introduction of domesticated livestock, we see certain groups of people starting to herd cattle, sheep, goats, and relying less on marine foods than in the past. And these are two contemporary depictions of people using cattle for transport. So the one on the left there is by an unknown Dutchman who visited the Cape before 1713. And it's a great little sketch. And on the right is one by Samuel Daniel from 1805. Has a much more European artistic style, but is still an accurate depiction of using cattle for transport. So, to better understand the lives of those 50 human skeletons found along the coast and figure out how they fit into our picture of the past, I've developed a Strontium 8786 isoscape, which is an isotope landscape map for the Western Cape Province, or at least most of the Western Cape Province. Yeah, like I mentioned, an isoscape is a spatial predictive model of strontium variation. And I've made that model based on strontium from wild plants. And most of this work is building upon work done by Copeland et al. In 2016 from the South Coast. So to create an isoscape, one needs a very large reference database of strontium ratios. So I've been driving around the Western Cape on little back roads, with some of my colleagues, and stopping where we can find wild, undisturbed plant growth growing on identifiable geology. And together with my colleagues, we've collected over 300 samples, trying to fill the gaps in the existing data. Each location, I will take GPS coordinates, take photographs, various descriptions of the geology, and in particular, take a small collection of plants, like you can see in the middle, and keep them in paper packets so that they don't grow mold, because that can be a problem. Back in the lab, I then cut up the plants into tiny little pieces, put them in small porcelain crucibles, and then ash them in a mussel furnace set to 650 degrees Celsius and leave it overnight. And the next morning, you find a small amount of ash in the bottom of the crucible, which is then homogenized, mixed, and weigh a tiny amount of that ash, dissolve that ash in acid, and separate or isolate the strontium out of that ash using ion exchange column chromatography. There's me in the lab, pipetting something. There's a whole process, different pHs and acids and all sorts of stuff. Then that isolated liquid strontium from that plant sample is then put into your mass spectrometer, and you get a ratio, right? Like I explained earlier on. And the problem with all of this whole process, is that it's long, and it takes a lot of time and a lot of money. It's expensive. And so researchers who look into this kind of thing try to predict strontium ratios as much as possible at unsampled locations. So you need some samples, but to reduce the cost and reduce the time, you model strontium ratios across your landscape. And this is where isoscapes come in, these predictive spatial models. So there are multiple different ways of creating isoscapes. I'm not going to go into too much detail with this. I feel like I've given you lots of detail already. But if you do have questions, we are welcome to chat about it. I basically used a machine learning random forest approach in a piece of software called R. And I incorporated these additional variables, like I mentioned in the beginning, geological age, but sea salt, dust deposition, all these additional factors that can influence your strontium ratio on the surface found in plants, and therefore animals and humans, etc. So here's my model. Here's my isoscape. The big picture on the left is the actual isoscape with all the sample points in red. And those are both the ones that I collected personally, as well as those that were previously published and publicly available. And on the right-hand side, so obviously, sorry, the color represents the dark purple to green, yellow represents the range in isotopic variance. From around 0.709 to 0.724. That's the range we see here on the Western Cape. And now this little diagram on the top right is an error map. So it's a map of the standard deviation at each point, and that tells us how confident to be in these predictions. So there's a range, but you can see that for most areas of the map, there's low uncertainty, and the map paying with high sample density. Now, obviously, areas that haven't been sampled are we can't be too confident, but most areas of the map are well sampled and have good uncertainty, so low uncertainty. Now, zooming in a little bit onto our area of interest along the west coast, where most of these 50 skeletons were found, the ones I've been talking about. I plotted the pre-2000 BP skeletons here. So these are the skeletons I'm calling the hunter-gatherers. We don't have actual evidence of that, but I'm calling them that because you can see that each of these individuals, exactly each of them. Let me just say that I have a little bit so they don't overlap. Some of them are found in pretty much the same place. So the locations are varied very slightly, so they can have in this distinct colors, and those colors are I'm using the same color gradient as the base map. And what's important is pretty much all of them, most of them, map very well with their surroundings. So the color of their dental strontium ratio maps pretty well to where they were found. And most of them were along the coast, but even the individual on the top, the light green individual, was found in the mountains there and has a much more terrestrial signal. So basically, people have values that map closely to where they were found. But then, shifting post-2000 BP, the group of individuals I call the herders to a much more diverse signal. So similarly, these are people found quite often along the coast, but we see much more terrestrial signals on people that were buried at the coast. And we also see people who have marine signals, see those dark purple colors, buried more or further inland. So basically what this means is that people were moving around in this period, right? So pre-2000s, or 2000 BP, we see a localized postal signal with probable, look, we can't exclude movement up and down the coast because that pretty much all has the same ratio. I'll show you again, right? This dark blue purple color is all pretty much the same. So we can't exclude that people moved up and down a little bit, but we can say quite confidently that people weren't moving inland. So there's a pretty clear pattern of multi-generational, long-term hunter-gatherer communities living in pretty much the same place, relying predominantly on marine resources, or at least foraging in the immediate vicinity of the coast. Then, like I mentioned, post-2000s, we see a shift with in this herder group, a much more diverse and more terrestrial signal, which means that people were moving around, there was increased mobility, there was increased consumption of foods from further inland. And this aligns a lot with other evidence for the arrival of domesticated animals to the Cape at around this period of 2000 BP. So I haven't really gone into that much, but we have fossil evidence. We have various evidence of the arrival of domesticated animals, and we can presume, and we can presume that this arrival of livestock caused a major shift in the economic, social, political, and quite obviously spatial organization of these coastal hunter-gatherer societies Uh, Thank you very much for listening. There's a bunch of people that have helped me with this research and funded it and made it possible. I've listed them there, as well as the core research team of Prof. Seeley, Prof. LaRue, Dr. Haley Corpora, and Karen Gray. Thank you very much. Are there any questions?
You mentioned a lot of kind of
Yeah, sheep, cattle, and goats. Look, I'm looking at the timing. So I haven't plotted all of these values you're seeing here on either plants for the isoscape itself or humans for the little circles. All I'm looking at is we have evidence for livestock arriving in the Cape at around 2000s. And if I, oh, okay, sheep, cattle, and goats. Yeah, as far as I'm aware. I am not an archaeologist by training. Big caveat. I have a background in forensic anthropology, but I have it on good authority. At around 2000, we see introduction of various livestock. Yeah. Question.
would be able to separate the goat livestock so they'll combine into single unit as people go. You wouldn't be able to do that in the goats might people come in before cattle. Yes, yeah, I think can you separate that?
I don't believe so. Possibly if we had better evidence of like some confirmation that a particular individual was really talented. But if we could do that, then maybe we could do better to our separate that those who can handle versus people. All of these skeletons don't have that sort of information. So all we know, based on their strontium isotopes, is that the herder group, the group I'm calling the herders, i.e. the more recent group, who were probably exposed to the idea of herding, they were moving around. That's all I can say. Yeah, I hope that answers your question. Anything else? Okay.
Since then you were looking mainly at like Strontium and alcohol, are you also compare actual individual to?
I haven't done that work, but it can be done. So I also didn't do extract something from these skeletons. That was done previously by Prof. Seeley and co. I only came in kind of recently and created this isoscape, trying to figure out what was going on with the individual strontium values. Like, what did they mean? You know, like we had a vague understanding, but this sort of map is helpful. But yeah, 100%. One could try and get ethical permission to go back and sample the same skeletons and sample their femurs, for example, ephemera, or rib bones, various tissues, and you can get a better picture of whether they were moving across the landscape and where they were moving to. So there are various statistical methods of, for example, and trying to range them to various parts along the landscape. It does get complicated because obviously if they're eating a diverse diet, if they're moving far and relatively quickly, there isn't a decent amount of time for a particular region's signature to be incorporated into their tissues. So it does get a bit tricky. Yeah. It depends on the tissue. So, for example, like I mentioned, dentition develops in utero and enamel doesn't remodel. So once your tooth enamel is formed, it's formed for the rest of your life. And that's why it's really interesting to, as a sampling tissue of choice because we get a picture of the individual as a child, or when they were born, and then first couple of years of life. Alternatively, one can sample like an M3, a third molar, which only forms much later. So then you can get a picture of closer to adulthood or like the end of the teenage years. And if, for example, like I mentioned, sampling a femur does remodel, your bones remodel, and they remodel at different rates as well. So a femur remodels more slowly than a rib, for example. So a sample from a femur will represent kind of a broad range of adult life, an average adult period, whereas a rib will represent a shorter period towards the end of that person's life. And if, obviously, it's kind of rare in archaeological contexts, but in modern forensic contexts, you can sample someone's hair or nail and get a much more recent period of their life. Yeah. But sorry, just going back to like as soon as you eat the food, it's there's going to be some calcium and strontium in it most of the time, and that'll just be incorporated into your diet and into your bloodstream and spread around and into your bones and stuff immediately. Yeah, there was a question here. Sorry.
Question was, how is the different diet being taken in influence the result of certain isotopes, depending on what they eat, and within that sample, you can tell is that isotopes there, or lesser, or is that numerous and influence?
Yes, I think I understand what you mean. That's a very good point. So, even for living stars, there's a lower concentration of strontium, and the plant star has a higher concentration of strontium because there's more calcium and therefore there's more strontium in the sunlight. That's one point. But the isotopic ratio doesn't depend on the concentration of the abundance. More depends on location. So even if you get a cow or herbivore grazing on a piece of land, if that piece of land all has a kind of uniform strontium availability, that's all right. Whether you eat the grass or whether you eat the cow, you'll get ultimately the same ratio. Does that kind of answer the question? I hope so. I believe so, yes. I didn't do any carbon dating, radiocarbon dating. No, this is Judy's photograph. I don't know if she did she wouldn't have done the dating herself, but she would have sent samples off for carbon dating. Yes, yes. Yeah, good point. We are currently writing up, not yet submitted, but yeah, in due course. Thank you. It's almost there. It's almost there. I have some final tweaks. I also have more plant samples to add to this model. So these models can constantly be refined. It's kind of an iterative process. So, but yeah, we're getting there. Thanks, Shelby. That's a great question. I don't think so. I don't think so. You could. But do you have a response to that? Or? Go ahead. No, that's a good point. I didn't really go into this, but if you are familiar with other isotope systems, most isotopes fractionate, meaning they like change in biological systems as chemical and metabolic reactions occur, and you get shifts in the ratios of heavy to light isotopes. Strontium doesn't. There's a very small amount of fractionation because the masses are so big and the mass difference is so tiny. There's a very little amount of fractionation, and we correct for it mathematically. So we can ignore fractionation in strontium isotope. Well, but carry on.
So I think maybe a better way to look at those kind of differences if you kind of say sheep, maybe graves. I'm not sure what sheep do, but if they make graves, if you're first to include more browse in their diet, then you would rather use stable carbon isotopes to distinguish those differences. But I think now, if you have a bone, you know how old it is, but you're not sure what animal it is, you probably want proteometers
Yes.
to distinguish it, probably the fastest way to get it to cattle or sheep. But if you know that there is a distinct dietary niche, then you can use the carbon isotopes to distinguish grazing from browsing.
That's a good point. And to a certain limited extent, one can possibly do something similar with strontium. Again, if you're confident that the animal species you're studying has a feeding niche. So grazing versus browsing is a good example because grasses usually have pretty shallow roots compared to shrubs and trees. So this isn't if, because it doesn't always occur, but if there are rooting depth specific differential strontium uptake into different plants, then you could like make an isoscape, one of these models, purely on grass samples or purely on shrubs, and then potentially get a different picture. Only browsers versus only grazers. There's potential for that, but it's complex. And my model couldn't do that because my model is created with kind of bulk plant samples, and that's to get a sort of average signal and not focus on one particular type of plant. I got 5 plants per sample and pooled them together. No, so I pooled them and got one strontium ratio out of that group of plants. The reason for that is cost, and also to specifically get an averaged sample for one location. And I targeted grasses, shrubs, and trees wherever I could in that group of 5, because most animal, I don't know about most, but humans have a broad diet, right? So they incorporate various things, and they'll eat animals, they'll eat various things. So I didn't want to hone in on one specific type of plant.
Last question, and I think everyone else can.
Yes, yeah.
The one problem with what I said with distinguishing grasses and berries is that it doesn't work the way in Western Cape because all the plants are seen to be. For some isotopes, but for common all plants are feeding retreat, but it's just due to high versus simplified.
If you had a growth this concentrated on the coast, you could have to travel very far and get there and really it's a political thing.
Yes, it's all
yeah, it's all on the coast and you have to do a lot of things. You step over that's it.
You can separate you can't distinguish from others but yeah, you could say that there were transhumans based on other isotopes like oxygen or
Sulfur,
or
potentially, and strontium as well. I mean, you can see that there's quite a big gradient between the colors. The problem is, so
You might have here, signal. It could be theoretically cut from out here, you go all the way down in that's it.
The isotope world is there's when you're incorporating things in your diet, there's various ways that particular signal can be created. You know, various combinations of plants and various combinations of locations that can result in the same strontium ratio or other isotopic ratio. The same pattern. I believe so. I believe we're almost at like saturation. Yeah. Obviously, there are gaps.
Farmland that we can't really sample. I do have one thing more here, and I do have one thing more here now. There are.
We could potentially do a better job, but I don't think the model is going to change much. Exactly.
And they check the Strontium and the whole thing, and the map they show was so manly.
Yeah. Yeah. Yes, yes. It really does depend on the geology, and in geologically complex regions, there'll be much more change, and the model will be much more diverse. And in more geologically complex regions, you'll need to collect more samples in order to overcome that. Otherwise, your model is going to be wrong, right? Now. No problem. No problem. What's this? How do I do it? We cancel that. You have a question?
Yes.
How do you?